So, let's check out the history of how clinical research evolved from anecdotal observations to the rigorous, ethically grounded, and safe science that we practice today. We'll explore the fundamental principles that protect participants. So, number one would be protecting the participants. The second would be to ensure scientific validity. Ensure the scientific validity and ultimately translate discoveries into treatments that save lives. So, this would be discoveries into treatments. So, this is the overview of what we are going to learn in clinical research, and let's begin. So, what is clinical research? Clinical research is a systematic investigation conducted with human participants. So, let's mention here that it is done on human participants to determine the safety and efficacy of medical interventions. These interventions can be drugs, devices, and procedures. This can also be behavioral interventions for people who are going through behavior changes. And it is also the critical bridge that connects basic laboratory science to patient care. Unlike preclinical research in animals or test tubes, clinical research involves real humans with real diseases, making it the gold standard for evidence generation. This is why clinical research operates under stringent ethical oversight and regulatory frameworks. So let's mention here that it operates under stringent ethical oversight and regulatory frameworks. Why do we do this? Because we are asking people to participate in studies where outcomes may not be certain. So, the outcomes may not be certain in these studies. So, we have a profound responsibility to protect them while advancing the medical knowledge. Let's start at the very beginning. Many don't realize that clinical trials have ancient roots. The earliest documented trial is in the book of Daniel in around 500 BCE, where dietary interventions were compared. But the modern era of clinical research truly began with James Lind in 1747. So, Lind was a Scottish Navy physician. So he was a Scottish Navy physician and he was really appalled by the devastating scurvy deaths. Right? And this was happening among the sailors. He conducted a comparative trial with different treatments. So some sailors received citrus, others received other remedies thought to cure scurvy during that time. This was revolutionary because he systematically compared the treatments under similar conditions. So, a comparative trial with different treatments. This was revolutionary because he systematically compared the treatments and the results that were given under similar conditions. He detailed documentation of methods and results, established the prototype for controlled clinical trials that we still use today. So, May 23rd is an important date and it is now celebrated. So, let's write it here, May 23rd, and it is celebrated as International Clinical Trials Day. This was done to commemorate Lind's groundbreaking work. Now, let's look at some ethical milestones. While Lind pioneered the method, history shows us that ethical oversight was often lacking. In 1900, Walter Reed conducted his famous yellow fever studies. But critically, he was one of the first to obtain written on informed consent from his participants. This was revolutionary because most researchers at that time didn't think they needed consent. However, it wasn't until after World War II that ethics became truly formalized. Now, while Lind pioneered the method, history shows us that ethical oversight was often lacking. In 1900, Walter Reed conducted his famous yellow fever studies. But critically, he was one of the first to obtain written informed consent from his participants. This was revolutionary because most researchers at that time didn't think they needed consent from the participants. It wasn't until World War II that ethics became truly formalized. Now, the Nuremberg trials exposed horrific Nazi medical experiments on prisoners, leading to the Nuremberg Code in 1947. This was the first international code stating that voluntary informed consent is absolutely fundamental. No exceptions was given. This single principle reshaped medical ethics globally and remains the cornerstone today. So let's look at the Nuremberg Code, which was a response to the Nazi medical experiments. It establishes voluntary consent as a fundamental right to the participants. Moving on, let's look at randomized controlled trials or RCTs. In 1943, the Medical Research Council in Britain conducted the first double-blind controlled trial of patulin for common cold, establishing the blinding methodology. But the landmark trial that really revolutionized clinical research was the 1946 streptomycin trial for tuberculosis. This trial was meticulously designed with systematic enrollment criteria, clear outcome measures, and randomized allocation, which is the first major RCT. So let's mention here that the trial was meticulously designed with systematic enrollment criteria, clear outcome measures, and randomized allocation. What does RCT mean? RCT stands for Randomized Controlled Trial. This is a study design where participants are randomly allocated to either receive the new treatment being tested or a controlled treatment. And the controlled treatment would be a standard therapy or a placebo. The randomization is crucial because it reduces the bias and ensures that the treatment and control groups are as similar as possible in all respects except for the treatment they receive. Bradford Hill, who led this work, proved that you could conduct rigorous, objective medical research without relying on physicians' subjective clinical impressions. So let's mention here that Bradford Hill led this work and proved that you could conduct rigorous, objective medical research without relying on the physician's subjective clinical impressions. This trial's methodology This trial's methodology has influenced virtually every area of clinical medicine for over 75 years. Moving forward, the World Medical Association established the Declaration of Helsinki in 1964. So let's mention here that it was the World Medical Association. It remains the foundation for ethical guidelines worldwide, updated regularly to reflect emerging concerns. However, one of the darkest chapters in American medical research, the Tuskegee syphilis study, continued until 1972, where African American men with syphilis were left untreated to observe disease progression even after penicillin became available. This unethical study, even after the Helsinki Declaration, led to the Belmont Report in 1979, which articulated three core principles: respect for persons, informed consent and protection of the autonomy. Second would be, so let's mention here that the respect for person includes informed consent and protection of autonomy. The second would be beneficence, which means maximizing benefits and minimizing harm. So, maximizing benefits and minimizing harm to all the participants that are participating in the study. And justice. Justice includes fair distribution of research benefits and burden. So, fair distribution of research benefits and burdens. These principles directly addresses the Tuskegee failures and remain the ethical foundation in many countries today, including the US. Let's move on to modern clinical research framework, or the ICH-GCP. Today, clinical research operates under what's called ICH, Good Clinical Practice Guideline, established in the 1990s through international cooperation between pharmaceutical industry, regulatory authorities, and academics. So let's mention here that it was established in the 1990s through international cooperation between pharmaceutical industries, regulatory authorities, and academics. Let me break down what ICH GCP actually stands for and what it does. So, ICH stands for International Council for Harmonization of Technical Requirements for Pharmaceutical for Human Use. And what is ICH? So, ICH is an international organization. It is an international organization that brings together regulatory authorities and pharmaceutical industry experts from major markets, which means the United States, the European Union, and Japan, to develop harmonized technical guidelines for drug development. So let's mention that it is there to develop harmonized technical guidelines for drug development. The goal is simple but powerful. It is to create uniform standards so that a drug developed and tested according to these guidelines can be submitted to regulatory agencies worldwide without duplication of testing. This saves time, reduces animal testing, and improves safety standards globally. Let's look at what is GCP. So, GCP stands for Good Clinical Practice, and it is a comprehensive set of ethical and scientific standards for designing, conducting, recording, and reporting clinical research. So, let's mention that it is there for designing, conducting, recording, and reporting clinical research. It's essentially a quality assurance framework that ensures clinical trials are designed, executed, and reported in a way that protects participant rights and safety while producing reliable, credible data. So, the ICH GCP E6R3 is the current version. Let's mention this. So, the ICH GCP E6R3 is the current version released in 2023. And it is the global standard that we use today. Let's see what IRBs do. IRBs stand for Institutional Review Boards. They are also called the Ethics Community or the Ethics Committee in many countries, ensuring protocol oversight according to ICH GCP standards. In the United States, the FDA or the Food and Drug Administration regulates clinical trials and has the authority to inspect sites for compliance. In Europe, it is the EMA or the European Medicine Agency, serves the same regulatory role. These aren't bureaucratic obstacles, they are critical safeguards. Modern clinical research has also embraced digital innovations like the electronic data capture system, decentralized trials where participants don't need to travel to clinics, and real world evidence from routine clinical practice. The entire field is evolving to make research more accessible while maintaining the highest standards of scientific rigor and ethical conduct according to the ICA GCP. Clinical research is built on four cornerstone principles, and I want you to really absorb these because they guide every decision in trial, design, and conduct. First is the ethical conduct. The participants' rights and safety always come before scientific interest or societal benefits. Always. No amount of potential medical advancement justifies harming a research participant. This principle emerged from the horrors of unethical research we discussed earlier and is now enshrined in international guidelines like the Declaration of Helsinki and the Belmont Report. Second is the scientific rigor. We must design studies that actually answer our questions with robust data. This means clear protocols, valid statistical analysis, and reproducible results. You cannot have sloppy science and call it research. The scientific rigor principle is why we have detailed statistical analysis plans and why we mandate randomization, blinding, and intention to treat analysis in RCTs. So let's mention here randomization, blinding, and intention to treat analysis in RCTs. Third is the informed consent. Participants must receive comprehensive, understandable information about what the study involves, the potential risks that comes with it, and the expected benefits with their alternatives. This information must be documented. Consent must be voluntary without coercion. Informed consent is not a signature on a form. It's an ongoing dialogue ensuring that the participants understand what they are agreeing to. The fourth and the last one is confidentiality. We have an absolute obligation to protect participant privacy and secure sensitive data. This includes using identification codes instead of names in databases, encrypting electronic data, limiting the access to authorized personnel only, and securing physical records. These four principles form the ethical and scientific foundation that distinguishes clinical research from exploitation. Let's get practical about what ICA GCP actually requires in day-to-day operations. First is the protocol-driven approach. Everything must be defined in advance. The study objectives, methods, statistical plans, everything. You can't modify your research mid-study based on what you are seeing. The protocol is your commitment to participants and regulators about how you will conduct the research. The second is a qualified personnel. Your investigators must be experienced. Your staff should be trained. You can't just assign someone to manage a clinical trial without proper training. The ICA GCP specifies the educational background and training requirements for different roles. A clinical coordinator managing participant visits, a data manager ensuring data quality, a biostatistician designing analysis. Each of these roles has defined competencies. The third is the IRB or Ethics Committee oversight. Protocols must be reviewed and approved before you enroll anyone. Your IRB must assess the risk-benefit ratio. Are the risks justified by the potential benefits? They review your informed consent form. Is the language clear? Does it adequately explain risks and alternatives? They identify vulnerable populations requiring extra protection. So, who are these vulnerable populations? So, this would be children, prisoners, and people who are severely ill. The IRB is not a one time gatekeeper, it's an ongoing guardian of ethical conduct. Fourth is safety monitoring. You must track adverse events systematically. If something serious happens, you report it immediately. You can't wait until the study ends. Serious adverse events or SAEs have reporting timelines measured in days, not weeks. Many trials have a data safety monitoring board. So let's mention here that it is a DSMB or Data Safety Monitoring Board, which is an independent committee that periodically reviews safety data and can recommend stopping a trial if unacceptable risks emerge. The fifth is quality insurance. Your study is subject to audits and inspections. You must maintain detailed documentation that proves you followed the protocol and operated ethically. Regulatory inspections can happen at any time, and when they do, they examine source documents, the original medical records, and compare them to what you recorded in your case report forms or the CRFs. So let's mention here that they look at the CRS or the case report forms. This is done to verify the accuracy. These aren't suggestions, they are requirements that sponsors and investigators must satisfy. Let's look at clinical trial protocol. The protocol is your blueprint for the entire study. It typically begins with scientific rationale, which means why are you conducting this research? What does existing evidence show? And why do we need this trial now? Then comes the study design. Are you doing a randomized control trial or the RCT, an observational study, or a crossover design where participants receive both the test treatment and control in sequence? This choice depends on your research question. Next is the inclusion and exclusion criteria. This precisely defines our study population. We have to be specific, which means age, range, disease stage, concomitant medications allowed or forbidden. This specifically prevents investigators from including inappropriate participants. So let's mention that we have to make sure of the age range, the disease stage, and the medications that are allowed or forbidden. Then comes the primary and secondary endpoints. What outcomes will you measure? Is it safety? Is it efficacy? Or is it quality of life? Your endpoints must be defined before data analysis begins to prevent what's called outcome switching. So let's mention that we have to avoid what's called the outcome switching, where researchers conveniently change which outcome they report based on what the data shows during that point. Finally, the statistical analysis plan or the SAP, including sample size calculation. How many participants do you need? What's your power calculation? Power is the probability that your study will detect a true effect if it exists. We typically want 80 or 90% power. So let's mention that the power should be between 80 to 90%. How will you analyze the data? Will you do an intention to treat analysis, which analyzes participants according to their randomized group, regardless of whether they completed treatment, or a pre protocol analysis, looking only at those who completed the treatment. All of this prevents bias and ensures that the results are credible and interpretable. So, all of these points mention if the study done is credible and interpretable. Let's look at the phases of clinical trials. Let me walk you through how drugs move from laboratory to patients through 4 distinct phases. So let's mention that it moves from the laboratory to patients in 4 phases. So the phase 1 typically includes 20 to 100 healthy volunteers and is purely about safety and dosage. So the main function or the motive of the phase 1 study is to focus on the safety and dosage. We are asking, is this drug safe? So let's mention, is this drug safe? Is what we want to know. What dose can people tolerate? How is it absorbed, metabolized, and eliminated? Which means the ADME studies of absorption, digestion, metabolism, and elimination. There is no exception of benefit in phase 1. The volunteers are usually healthy people receiving payment to contribute to science. If phase 1 goes well, we move to the phase 2. Phase 2 involves 100 to 300 patient volunteers, people who actually have the diseases that we are studying. Now you're asking, does this drug work? What are the side effects? And what is the optimal dose for efficacy? So let's mention that we want to know if the drug works. What are the side effects? And what is the optimal dose for efficacy? We are collecting preliminary evidence of effectiveness while watching carefully for adverse effects. So we have to look for adverse effects during this stage of the process. Moving on to the phase 3, which is a pivotal trial. This is what regulatory agencies scrutinize most carefully. With over 1,000 to 5,000 patient volunteers, we are confirming efficacy, comparing to standard treatments or placebo, and gathering comprehensive safety data. Phase 3 trials are typically RCTs or randomized controlled trials because randomization provides the most robust evidence. If phase 3 shows that our drug is safe and effective, we apply for regulatory approval. In the US, we submit a new drug application. So let's mention that in the US, we submit a new drug application or the NDA. This goes to the FDA, which is the Food and Drug Administration. In Europe, we submit it through EMA, which is the European Medicines Agency. Finally, the phase 4 is the post-marketing surveillance. This continues after the drug is approved. This can involve large observational studies monitoring long-term safety in broader populations. Phase 4 often reveals rare adverse effects that weren't apparent in the controlled phase 3 setting because phase 3 typically enrolls thousands of participants, while phase 4 may eventually involve millions of users. So let's mention here that it includes millions of users. Now, data quality is absolutely critical. Your conclusions are only as good as your data. We implement multiple safeguards for this. So, the source data verification means that we go back to original medical records, the actual clinical notes, the lab results and the imaging reports, and compare them to the data entered in our case report forms or the CRFs. This catches the transcription error. So we really have to focus on the case report forms. Case report forms are standardized instruments. Every site, every participant answers the same questions in the same way, ensuring consistency. The electronic data capture or the EDC system, now standard in most trials, allow real-time data entry with built-in validation checks. If a value seems impossible, like a 200-year-old person, the system flags it. So let's mention an example that if it shows a 200-year-old person, the system would flag it and we would know. We maintain complete audit trails showing who entered data, when they entered it, and if they made any corrections, all time stamped and documented. Finally, quality control includes regular audits where monitors visit sites, spot check data, and verify accuracy against source documents. This infrastructure might seem burdensome, but it's what separates credible research from unreliable data. Let's move on to biostatistical methods. So, biostatistics is fundamental to clinical research design and analysis. Sample size calculation determines how many participants we need. This isn't arbitrary, it's based on the effect size you expect to detect and the confidence with which you want to detect it. Too small a sample and you won't have the power to detect a real effect. Too large and you're wasting resources and potentially exposing unnecessary people to risk. Randomization is randomly assigning participants to treatment groups. It is powerful because it balances known and unknown confounding variables. If you just let doctors choose who gets the new treatment, you introduce bias. With randomization, treatment and control groups are comparable. Blinding is where participants and sometimes investigators don't know which treatment is being given. This prevents bias in patient perception and investigator assessment. In a single blind design, participants don't know their assignment. In double blind, neither participants nor investigators know. This prevents the placebo effect from influencing the result. So let's mention here that in a single blind design, the participants don't know their assignments. In a double blind, neither participants nor the investigators so The intention to treat analysis maintains the benefits of randomization by analyzing participants in the groups they were randomized to, even if they didn't complete the treatment. This preserves the effect of randomization. And finally, the statistical significance testing gives us the p values. So, it gives us the p values and the confidence interval, also the effect sizes. Finally, the statistical significance testing gives us the p values, the confidence intervals, and the effect sizes. The quantitative language for expressing how confident you are in your results. A p value of 0.05 means there's a 5% probability that the observed result occurred by chance. All of these methods exist because humans are prone to bias, and these statistical approaches help us to overcome it. Let's look at the study designs. There are multiple research designs appropriate for different questions. The randomized controlled trial is considered the gold standard when your goal is to evaluate whether an intervention causes a specific outcome. So, when an intervention causes a specific outcome. Random allocation to treatment and control groups eliminates selection bias, and if you have enough participants, balances confounders. However, RCTs are expensive, time consuming, and ethically impossible for some questions. So they are expensive, time consuming, and ethically impossible. For some questions. What if your question is, does smoking cause lung cancer? You can't randomize people to smoke. For such questions, observational studies are appropriate. Cohort study follow people with and without the risk factor forward in time to see who develops diseases. You start with non-diseased people, identify their exposure status, and follow them prospectively to see who develops the outcome. Now, cohort studies follow people with and without the risk factor forward in time to see who develops diseases. You start with non-diseased people, identify their exposure status, and follow them prospectively to see who develops the outcomes. So, in these cohort studies, we start with non-diseased people and identify their exposure status. Case control studies start with people who have the disease and those who don't, then look backward at their exposures. This is efficient for rare diseases. Cross-sectional studies capture a snapshot at one point in time, measuring both exposure and diseases simultaneously. Each design has different strengths and weaknesses. It's about matching the design to your research questions. Now, we all know that safety is paramount. Safety is very important. We define adverse events as any untoward medical occurrence in a trial participant. And untoward is key. So let's mention that this is the key here. It doesn't have to be caused by the study drug, but if it occurs during the study, we do report it. Now, serious adverse events or the SAEs are those that threaten life, require hospitalization, and result in disability, or they are medically important, which demands immediate reporting to sponsors and regulatory authorities, sometimes within 24 hours. So let's mention. We use standardized grading systems like the CTCAE, which is the common terminology. So, the common terminology criteria for adverse events. This grades the severity from 1, which is mild, to 5, which is death. So 1 is mild and 5 is death. Causality assessment determines whether an adverse event is likely caused by the study intervention or something else. You might use a probability scale which includes definite, probable, possible, unlikely, or unrelated. So it would look like this: definite, probable, possible, unlikely, and unrelated. This is the probability scale that we can use. Now, pharmacovigilance is systematic monitoring for safety signals, which are patterns suggesting an unexpected hazard. If 5 participants suddenly develop kidney problems, that's a signal that demands investigation. A data safety monitoring board is independent from the research team, periodically reviews safety data, and can recommend stopping a trial if unacceptable risks emerge. No one benefits from a trial that exposes participants to unacceptable harm. Now, a very important topic is ethical oversight. Ethical oversight is ongoing, not just at the beginning. The Institutional Review Board or Ethics Committee in many countries review your protocol before you start and must approve it. They assess the risk benefit ratio. Are the risks justified by the potential benefits? They review your informed consent form, which means is the language clear? Does it adequately explain risks and alternatives? They identify vulnerable populations requiring extra protection, which means children, prisoners, and the severely ill. The informed consent form is critical. It must be comprehensive yet understandable. Participants must understand what is the study about, what will they be asked to do, what are the risks, what are the potential benefits, what alternatives exist, how is their privacy protected, what happens if they are harmed, and can they withdraw the study. The informed consent form is critical. It must be comprehensive yet understandable. Participants must understand what the study is about. What will they be asked to do? What are the risks? What are the potential benefits? What are the alternatives that exist? And can they withdraw from the study? After approval, IRBs conduct ongoing review. Usually, it goes annually. It ensures the study continues to protect the participants. If serious adverse events occur, you must report them to the IRB. If you want to modify the protocol, you must get IRB approval first. This framework has evolved specifically to prevent exploitation. It is why researchers cannot unilaterally decide to experiment on humans. Clinical research is evolving rapidly to address practical challenges and incorporate technology. Decentralized trials reduce participant burden. Instead of frequent clinical visits, participants may do home based assessments. Some sites conduct visits virtually. This expands access, especially for geographically remote participants or those with mobility limitations. Real world evidence comes from pragmatic trials conducted in routine clinical settings rather than tightly controlled research environments. Instead of asking, does this drug work under ideal conditions with select patients, we ask, does it work in real practice with diverse patients? Patient centered outcomes reflect what matters to patients, which means quality of life and functional ability. Diversity initiatives ensure clinical trials represent the population who will use the treatments because drug responses can vary by ancestry and genetics. Finally, digital health integration are wearables, mobile health apps, remote monitoring provides continuous real time data. Replacing episodic clinical visits, these innovations aim to make research more efficient, inclusive, and participant friendly. So let's mention here that the digital health integration makes the research more efficient, inclusive, and participant friendly, while maintaining the scientific rigor.